Hello everyone, chapter 4 is all about the role of microorganisms in soil fertility. So under this, the topics would be decomposition and metabolism of organic compounds, nitrogen transformations in soils, and transformations of phosphorus, sulfur, iron, and manganese. Decomposition of organic compounds. So what is organic compounds? So this is any of the chemical compounds in which one or more atoms of carbon are covalently linked to atoms of other elements. So in short, organic compounds contains the carbon that is covalently bonded with another element. The composition of green plant tissues, this include water, which is about 75%, dry matter of 25%. And its elemental composition, it consists of carbon of 42%, oxygen 42%, hydrogen 8%, and ash 8%. Okay, if we are going to look at the chemical structure of these organic compounds, we have here glucose as an example. Carbon is always present, and it is covalently bonded with other elements. So here, carbon is covalently bonded with hydrogen and then oxygen, or with hydroxyl. So this compose the organic compounds. And we have here examples are the types of compounds: cellulose, which comprise 45% of the plant or any organic molecule. Hemicellulose 18%, protein 8%, sugars and starches 5%, fats and waxes 2%, lignin of 20%, and polyphenols of 2%. The rate of decomposition of organic compounds varies. So decomposition, this is the breakdown of large organic molecules into smaller, simpler compounds. So from sugar, starches, and simple compounds, these are rapidly uh, decomposed. And then as it goes down, the decomposition becomes slow. So from sugar to crude proteins to hemicellulose to cellulose to fats and waxes, and then lignins and phenolic compounds. So notice here that fats, okay, correlate natin with humans. These are not easily broken down. Okay, so what would be the reaction that takes place when organic tissue is added to aerobic soil? So there would be enzymatic oxidation of carbon compounds to produce carbon dioxide, water, energy, and decomposer biomass. And decomposer biomass is when microorganisms start to accumulate. There would be the release and/or immobilization of the essential nutrient elements such as nitrogen, phosphorus, and sulfur by a series of specific reactions that are relatively unique for each element. So this will be discussed in the last part of this chapter. Under nitrogen transformations and then assimilations of nutrient elements. And there would also be the formation of compounds very resistant to microbial action, either through modification of compounds in the original tissue or by microbial synthesis. Let us take a look at the decomposition that happens in aerobic soil. And this basic reaction accounts for most of the organic matter decomposition, as well as oxygen consumption and carbon dioxide release from the soil. So in organic soil, the carbon and the hydrogen-containing compounds or the organic materials are being oxidized because of the presence of oxygen. And also, in the presence of the enzymatic actions by the microorganisms, and this will release the carbon dioxide, water, and energy, including the accumulation of decomposer biomass. Now, um, in this process, mineralization also occurs, and that releases the nutrient elements from the organic compounds that have been decomposed. So the form that is absorbable by plants are released through the mineralization. So therefore, in this aerobic decomposition, it helps in the release of the essential nutrient elements such as the nitrogen, phosphorus, and sulfur, and including inorganic ions such as ammonium, nitrate, and sulfate. The decomposition in aerobic soil is different from the decomposition that happens in anaerobic soil. So, in anaerobic soil, the decomposition is very slow, and these are acted by a special microorganisms called methanogenic bacteria, including archaeas. So these are the ones that are involved in the anaerobic decomposition. So in anaerobic decomposition, the propionate or the propionic acids, these propionic acids are the short chain fatty acids that is a component of organic material in the presence of water and also the bacteria that is involved in anaerobic decomposition to produce acetate or acetic acid, which is an alcohol, plus carbon dioxide and methane. And these are gases, these two are gases, so they would escape in the atmosphere. Now, the process will not stop there because this acetic acid will again be acted upon by another bacteria, producing carbon dioxide and methane. So again, there will be the release of these gases. And then to continue, because there is carbon dioxide, it will react with hydrogen ions that are commonly present in the soil. So again, it is acted by microorganisms to produce water and methane. So there is the production of gases, which are the carbon dioxide and then methane, and also the alcohol, the acetate. Thus, yung produced product ng mga ito, they produces foul odors and also plant inhibitors. That is why in anaerobic, the decomposition is really slow. And also, the organic materials are only partially decomposed, not fully, unlike in aerobic decomposition. Now, there are factors that controls the rate of decomposition and mineralization. And when there is decomposition kasi, there will be a release of minerals, and that is what mineralization means, the conversion of organic compounds into inorganic compounds. So we have here environmental conditions in the soil. The pH, so it should be in the near neutral because most of the microorganisms, especially bacteria, thrive near neutral pH. So, and we all know that the abundant microorganisms in the soil are bacteria, and these are sensitive microorganisms, unlike fungi that they could thrive at any pH range because those are versatile microorganisms. Also, moisture, it should be sufficient, good aeration, and then warm temperature of 25 to 35 degrees Celsius. And this is the temperature requirement for most mesophilic or for mesophilic microorganisms. And most of the microorganisms are mesophilic. Iilan lang naman yung mga thermophilic microorganisms or yung mga microorganisms that could survive extreme temperature conditions. So in addition, is the quality of the added residues as a food source for microorganisms. So physical conditions of the residues include the size and the age of the residues. So sa size kasi, of course, parang sa composting lang yan na if you will shred the organic materials, the decomposition is faster. Okay? Sa age din, the younger the organic material, kapag green yan, di ba, the faster the decomposition. Pero kapag nag-brown na, because it has ano ba yan? Formed lignin and polyphenol contents, and these are chemical composition that have slow decomposition. And as the plant age, they will form this kind of compounds. Okay, next is the CN ratio. This is the carbon-nitrogen ratio, which means the ratio between the mass of the carbon to the mass of the nitrogen. Okay, so kapag young ang plant din, ang mga organic materials, they have low CN ratio. Now, kapag naging old din sila, they have higher CN ratio. And the rate of decomposition when it comes to CN ratio is the lower the CN ratio, the faster the decomposition. For example, we have yung sunflower or alfalfa, they have low or sakto lang na CN ratio, 25:1. Versus the CN ratio of hays or yung mga rice straw, which aabot ng mga 90:1. So yun, with that, which decompose faster? Of course, yung organic material with a low CN ratio, okay, which is the alfalfa and the sunflower. Now, okay, parang nawawala tong greenhouse effect dapat pala after that, decomposition. Okay, so greenhouse effect is the entrapment of heat by upper atmosphere gases such as carbon dioxide, water vapor, and methane, just as glass traps heat for greenhouse. Okay, so decomposing organic material in anaerobic conditions by microbes in the absence of oxygen releases methane into the atmosphere and even carbon dioxide. Now, methane is 26 times more potent or toxic than carbon dioxide as a greenhouse gas and is a significant contributor to the global greenhouse gas emissions. So, it increases the quantities of these gases in the atmosphere, and that will result to the global warming that may have serious consequences to mankind. So, one factor is this anaerobic decomposition because of the production of carbon dioxide and especially methane. Metabolism of organic compounds. So when we say metabolism, this is the biological oxidation of organic compounds by microorganisms to yield energy and another organic or inorganic compounds. And this is necessary for microorganisms because during that metabolism of organic compounds, they obtain carbon and energy for growth. They need these organic compounds also because these are also co-substrates in converting them into products such as carbon dioxide, water, and to increase their biomass or simply for them to grow and develop. Cellulose is probably the most abundant polysaccharide on Earth. And these polysaccharides are organic molecules with long chains of monosaccharides linked by glycosidic bonds. Examples are cellulose, starch, and chitin because they contain the glucose. Although in cellulose, uh, it is a polysaccharide with glucose as its monomer, while in starch, it is a straight chain polymer of glucose, and the glucose in cellulose are linked by beta glycosidic bonds, while the glucose in starch are linked by alpha glycosidic bonds. Now, cellulose, this is the major structural material of which plants are made of. And wood is largely cellulose, cotton, and paper are almost pure cellulose. Cellulose also has more hydrogen bonds between glucose units, making it a tougher fiber than starch. This is why wood is so tough. Okay, so the initial chain breaking step in cellulose decomposition requires the activity of specialized microorganisms that produce the enzyme cellulase. Okay, so the microorganisms must contain the enzyme cellulase in order for them to carry out metabolism of cellulose. Now, a cellulase, this is an enzyme no, that acts as a catalyst in living organisms, at which when we say catalyst, it will speed up the rate of a chemical reaction. And the chemical reaction we are talking about here is the metabolism. And in every organic compound or in every organic molecule, a microorganism can only metabolize this if they have this specific enzyme. So for cellulose, we have the cellulase. And these cellulase are components of a specific microorganism also. Okay? So the long chains are broken down by specialized microorganisms which contain the cellulase into short chains, then into individual sugar or glucose molecules which many microorganisms can metabolize. So the first step here is that the breaking of the long chains by microorganisms which contains the cellulase enzyme. Then after that, pag naging short chains na sila, then other microorganisms can now metabolize this cellulose. But without the first step, okay, there will be no decomposition of that organic material, or there is no metabolism of cellulose. Okay, most animals, with the exception of cockroaches, cows, and some other creatures, cannot digest cellulose. So, yan na, yung cellulose, it is bonded by B-glucose. Next one is starch. So, these are straight-chain polymer of glucose, while yung cellulose, a while ago, they are monomer of glucose. Glucose polymers in which glucopyranose units are bonded by alpha linkages. Okay, yung cellulose again, the glucose are bonded by beta linkages. Plants manufacture starch by bonding many glucose molecules end to end as a means of storing energy. Potato, rice, wheat, and maize are sources of starch, and these are considered in storing long-term energy and can be released when only needed. Now, what are the components of starch? Starch is made up of a mixture of amylase, around 15 to 20%, and amylopectin of about 80 to 85%. Amylose consists of a linear chain of several hundred glucose molecules, while amylopectin is a branched molecule made up of several thousand glucose units. And in every chain, it contains 24 to 30 glucose units. So if we are going to look at this illustration here, the amylose here is very simple. while the amylopectin, it is more complex. How about the metabolism of starch? The starch, these are insoluble in water, and they can be digested by hydrolysis. And hydrolysis, this is when the water breaks the chemical bonds of the organic molecules, and it is catalyzed by enzymes called amylases, which break the alpha linkages or the glycosidic bonds. Humans and other animals have amylases so they can digest starches. Animals and humans, we can digest starch by breaking it down into glucose and using the sugar as source of energy. Lastly, we have chitin. So the formula for chitin is C8H13O5 nitrogen. So chitin is a long chain polymer of nitrogen acetylglucosamine, which is a derivative of glucose. It is the main component of the cell walls of fungi, exoskeletons of arthropods such as crustaceans, examples are crabs, lobsters, and shrimps, and also insects. This chitin is the component of the radulas of mollusks and the beaks of cephalopods, including squid and octopuses. So if you are going to think of the chitin, this is the component of the hard shells of any crustaceans. Okay, so for chitin metabolism, they are biodegradable in the natural environment. So, yung decomposition nila is can occur in the natural environment. The breakdown may be catalyzed by an enzyme called chitinase, and this is secreted by microorganisms such as bacteria and fungi and produced by some plants. Some of the microorganisms have receptors to simple sugars from the decomposition of chitin. Now, if chitin is detected, they then produce enzymes to digest it by cleaving the glycosidic bonds in order to convert it into simple sugars and ammonia. So in here, pansinin niyo in the metabolism, it is breaking down the bond. Tapos, when that bond is broken down kasi, that compound or complex organic molecule become a simple organic molecule. And from that simple organic molecule, they can now convert it into simple sugars, which they use as energy. Hemicellulose and pectin. So these contain a number of sugars. In the hemicellulose backbone, it contains the sugars xylose, mannose, and galactose. In the hemicellulose side chains, it contains the sugar glucuronic acid and galactose. So hemicellulose is a polysaccharide consisting of shorter chains, 500 to 3000 sugar units. It is embedded in the cell walls of plants. Some hemicellulose are water-soluble and some are water-insoluble. Next is pectin. These are methylated esters of polygalacturonic acid which consist of chains of 300 to 1000 galacturonic acid units joined with alpha 1 alpha to 4 linkages. This galacturonic acid is an acidifying agent and it is the monomer of pectin molecules. So, kung ang cellulose, ang starch, cellulose, ang monomer niya ay glucose, for pectin, it is the galacturonic acid. Okay. Pectin forms part of the primary cell wall of the plants and part of the middle lamella between plant cells where it helps to bind cells together. So, I think you have heard of the pectin already because the white portion of the rind of lemons and oranges contains approximately 30% pectin. And during ripening, pectin is broken down by the enzyme pectinase and pectinesterase. In this process, the fruit becomes softer as the middle lamella breaks down and cells become separated from each other. So here in this illustration, okay, so the galacturonic acid here is, okay, ayan, H-galacturonic acid. Let us move on with proteins, amino acids, purines, and pyrimidines. So let's start with protein. Proteins, these are complex biological substances composed of carbon, nitrogen, hydrogen, oxygen, and sometimes sulfur and phosphorus. It consists of polypeptide, which is a single linear polymer chain of amino acids bonded together by peptide bonds. So, you know, ano to? Protein is a polymer of amino acids. When we say kasi polymer, to differentiate pala polymer from monomer, polymer, it is composed of large molecules that are multiples of simpler chemical units called monomers. And monomers, these are small molecules bonded together to form a more complex structure called polymers. So polymer, these are combination of monomers. And monomers, when they are bonded together, they are called polymers. Okay? Digestion breaks protein down into amino acids because protein is they are polymers of amino acids. The breakdown of proteins. Plant proteins succumb to microbial decay, yielding not only carbon dioxide and water but also glycine and cysteine. In turn, these nitrogen and sulfur compounds are further broken down. So yung mga glycine and cysteine, these are nitrogen and sulfur compounds. And eventually, yielding inorganic ions as ammonium, nitrate, and sulfate, which forms the available for plant nutrition. Amino acids are the monomers that makes up protein. Every amino acid has a common structure, in which a central carbon is covalently bonded to a carboxyl group or yung COOH, an amino group NH2, and a hydrogen, and a variable R. Now, amino acids, these are molecules containing an amine group, yung COOH, a carboxylic acid group, and a side chain that is specific to each amino acid. Now, the key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen. The generic formula is H2NCHRCOOH, where R is an organic constituent. The amino group is attached to the carbon atom, here, immediately adjacent to the carboxylate group, the alpha carbon, carboxyl group. Okay? Amino acids are the structural units that make up proteins. They join together to form short polymer chains called peptides or longer chains called either polypeptides or proteins. These polymers are linear and unbranched, with each amino acid within the chain attached to two neighboring amino acids. So the process of making proteins is called translation and involves the step-by-step addition of amino acids to a growing protein chain by a ribozyme that is called a ribosome. So let us take a look at the structure of amino acids, peptide, and protein. So these are the amino acids, no? Now, amino acid plus amino acid is equal to peptide. Peptide plus peptide is equal to protein. So that is how amino acids, peptide, and protein are closely related. Now, for amino acid catabolism, the first step is the removal of the nitrogen or the amino group. So the reaction goes like this. Amino acid in the removal of amino group will form the ammonia. And this ammonia, with the addition or protonation of hydrogen ion, will form urea. And urea is excreted in the urine. So in here, we are talking about the metabolism of microorganisms. Okay, purine and pyrimidine. These are the building blocks of nucleic acid. So for purine, under it are the adenine and guanine. And then for pyrimidine, the cytosine, thymine, and in addition to that is the uracil. So uric acid is a metabolic end product of purine metabolism. Lignin. This is a complex polymer which is the building blocks are phenolic compounds. They are very large and complex. They consist of hundreds of interlinked phenolic ring subunits. The linkages among these structures are so varied and strong, only a few microorganisms, like the white rot fungi, can break them down. Decomposition proceeds very slowly at first and is generally assisted by the physical activities of the soil fauna. Now, lignin is one of the organic compounds that are not easily decomposed. So they are linked very strongly. Lignin, lignin peroxidase or ligninase is a hemoprotein from the white rot fungus Phanerochaete Chrysosporium with a variety of lignin-degrading reactions, all dependent on hydrogen peroxide to incorporate molecular oxygen into reaction products. Lignin is one of the most slowly decomposing components of dead vegetation, contributing a major fraction of the material that becomes humus as it decomposes. Lastly, we have aromatic compounds, and they are also known as aromatics or aroms. And this component of an organic material is what it gives them their aromas or odors. Typical aromatic compounds are benzene and toluene. A key step required for degradation of aromatic compounds is the formation of cleavage in the aromatic ring. So once there is a formation of cleavage here, then continuous na yung degradation ng aromatic compounds na ito. But this step is carried out by organisms or microorganisms that contain the oxygenase enzymes with a molecular oxygen as a reactant. So we'll end it here. The nitrogen transformations will be discussed in the next meeting, and that would be after your midterm exam. Okay,